Today we're going to talk about designing a results section. Now you'll notice that I've intentionally called this designing a results section, not writing a results section, because there is more to a results section than just writing. We've talked before about how a scientific paper should tell a story, and nowhere is that more true than in the results section. So when you're thinking of the results section as a story, you want to think of it kind of like a picture book rather than a novel. Because in a results section, it's really important to have both words and what I'm going to call illustrations. They may not necessarily be pictures, but they will be non-text things that help add to the story. So what do I mean by illustrations? There are many things you could do to illustrate your results. Obviously, you can use a photograph if you have something really cool that you have a picture of. That's probably more biologists than chemists like me. You could have a diagram. That's also probably more for biologists if you're looking at morphological features or something like that. You could have a table with data in it. Or you could have a graph. Now, which one of those you use depends on what it is that you're trying to illustrate. And we'll talk a little more about that later. So what you're going to do is you're going to start by picking your illustrations. Not by writing the text and then illustrating it, but by picking the illustrations first. And then only when your illustrations are in place are you going to explain each illustration with a paragraph or so of text. And those paragraphs are basically going to tell the story of each illustration. And so there are people that say that the proper way to write a paper is to start by creating your figures and then print them out, post them on the wall, and have other people come by and see if they can deduce what the story of the paper is just from the figures. So what you're going to do is build your results section around the figures. You cannot, however, just put figures in your results section, which is actually probably the most common error that I see when I have students do formal lab write-ups that are trying to parallel a scientific paper. You need to talk the reader through the story that the figure is telling them. Yes, there will be a caption that is separate from the text, but the caption is only an explanation of things like what are the axes and stuff like that. So it's not the whole story. Results sections should start with raw data. Now you see I put quotation marks in there, and that's not me just throwing quotation marks out randomly as some people do. What I mean by this is data that has already been translated into whatever units you were measuring at the time you were making your measurements. So if you've watched my video on Introduction to Chemical Analysis, you'll probably remember that the readings we get from instruments are rarely in the units that we want to use. If you are doing a colorimetric method, you're going to be measuring absorbance. You can transform that absorbance data into concentrations. How do you do that? Using your calibration curve. So when I say raw data, what I mean is the original data that you were looking at when you were trying to figure out what everything meant. I don't mean raw data without the quotation marks. True raw data is your voltage reading, your absorbance reading, okay, and that's not what I'm asking you to put in there, right? What you want is the data that's already been translated into the units you want to use. Once you have all that raw data in front of you, what you need to do is decide what sort of illustration you want to use. So a lot of times what you're going to be using a photograph for or a diagram is to show structural or morphological features. You'll use a graph if you want to show a trend or a relationship between two things. Generally speaking, you use a table if there's too many factors to put on one graph and you want to see all of those things compared at the same time. You could put like a whole array of graphs, and I've definitely seen people do that, but sometimes that's easier to see in table format. So once you've picked your illustrations, you're going to write that paragraph about each of your illustrations. Now, even though you have just written a brilliant method section, I hope, it does not hurt to start each paragraph with a brief summary of the method that you use to get those results. And the reason I say that is because your method section may have many different methods that were used to get a variety of results. And so you need to remind your reader how it was that you got the particular results you're talking about right now. When I say brief, I mean a sentence, right? You're not going to write it all out in gory detail the way that you did in the method section. That's what the method section is for. This is just a reminder. Hey, you remember how we used that colorimetric method to determine silicate concentrations? Well, here are the results that we got from that. Once you've explained how you got the results, then you're going to talk about what those results mean. And so you might do things like explain how things changed with time. This is also the place where you're going to include non-numerical data, the sort that can't be graphed. As the bacterial concentrations increased, we noticed a color change taking place in the flask. That's all well and good, and I could tell people to do this, and they still don't quite understand what I'm getting, because a lot of times what I see is I see people then taking all the data points that went into the graph and just recounting them in textual form. Our concentration was 1 micromolar at time 0, and then it increased to 2 micromolar at 30 seconds and 3 micromolar at 60 seconds. And that's a lot to know. That's what the graph is supposed to tell you. So you don't want to rehash the data that you just put in your table or graph. Remember, we're going to talk about general trends, relationships. So what you could say, for example, is the concentration increased linearly at a rate of approximately 2 micromolar per minute. Something like that. So that's a general trend. Okay. That trend might be on your graph. It might be that you have a linear fit on that, and it shows you what the equation for the line is, right? But that's fine. General trends, general relationships. So you don't want to actually just spell out all the data. You want to talk about generalities. You also don't want to put the data in a larger context. Okay? That's discussion section stuff. So you don't want to say, this was 5 times as fast as what was seen in a previous study by Chowdhury et al. So those are general guidelines. Now, of course, what we're going to do is we're going to go back and we're going to look at our 2 papers, the Rowe et al. 2012 and the Lynch et al. 2015. So let's do that. Let's start with the Rowe. So here's the beginning of the results section. And you'll see there are subheadings within the results section. That's a really good idea if you're looking at more than one thing. And you'll see that they start out by talking about what they did briefly. There is one sentence. Non-azenic trichodesmium cultures at high and low iron were used to generate the growth curves shown in figure 1. That's a brief rehash of what they said when they talked about how they grew things under low and high iron concentrations in the method section, which you can go back and look for. And then if you look at what's actually written here, they talk about specific growth rates that they observed. Now, that paragraph is describing what happens in figure 1. So figure 1 is the first illustration. And you can see what's in the figure caption, first of all. Figure 1 tells us what this is, trichodesmium growth curves with iron high, so the high iron concentration, and they tell you what was added, and the low iron concentration cultures. Okay, and so they're telling you which of these are which. Growth curves are shown in relative fluorescence units versus time, normalized to time 0. So they're telling you about the axes. Okay, it doesn't tell you what is happening in this graph. That's what's in the paragraph. This is just giving you the little bits of information so you can look at this graph, you can understand what each of those symbols are, and you can understand, for example, what are the error bars, how do they calculate those error bars. Just very basic information so you can interpret the graph. What the graph actually tells us was written in the paragraph. So you see here, what they're graphing is they're graphing a trend with time. How is the culture growing over time? Now, if you look on the next page, you'll see that there's a table of physiological parameters. Here we have 2 different cultures, the high iron and the low iron. And you'll notice that there are, let's see, trichome length, cell width, picograms of chlorophyll A per cell, FV over FM, and cells per mil. So 6 different parameters for these 2 cultures that they're comparing. If you think about how you would try and illustrate that in a graph, that would be kind of hard. You might be able to do a bar graph, but because these are all on very different scales, that might be pretty hard. Here, a table is an easier illustration to use because you can quickly compare that, for example, there are 3 times as many cells per mil in the high iron and the low iron culture, etc. There are a couple of paragraphs here that talk about the differences between those 2 cultures. Trichodesmium iron uptake experiments. Okay, so now we have another set of experiments. Here they actually have 2 sentences that describe very briefly how they did the iron uptake experiments. If you want more information, you go back to the method section. They talk about how they calculated the iron uptake from their measurements. And then here's the first illustration. We have the trichodesmium iron uptake for low iron in the light and low iron in the dark. Again, what are they comparing? FeCit, FeCl3, FeDFO, FeAero. So what these are these are different sorts of iron compounds, iron citrate, iron chloride, etc., etc. And these other 2 are bigger ligands that I don't remember the names of. Again, here you have multiple factors that would be really hard to display all of them in a graph. So they're put in a table. And because they're talking about how trichodesmium grows, then they have to put in some pretty pictures because we like pretty pictures. So they have SEM, they've got DAPI stain cells. All right, now we have iron uptake experiments with representative bacteria strains. The 2 trichodesmium-associated bacteria strains were supplied with various iron sources after acclimation to PC media where iron was limiting nutrient. That's methods. The iron-limited culture had a growth rate of 0.11 per hour through 36 hours. Okay, do you see what that is? Suddenly we switched over to here's a result. Here's a general trend. Over 36 hours, I'm sure they didn't just measure it at 0 hours and 36 hours, right? They probably have a whole growth curve, but they're just reporting a trend. Here's the iron-limited growth curves. You probably saw that coming, right? Because they talked about a general trend, okay? And do you remember 0.11 per hour for the first 36 hours? Microcilla marina was the one they were talking about. This peak point here is about 36 hours, looks to me. So there's a steep growth and then it levels off, right? So they're talking about here's what we see. Fairly quick growth for the first 36 hours. They have just told us what we see in the graph. That's sort of what we're looking for. So you can go through and you can look at more. There's a lot more growth curves and whatever in this paper. But what I want to do is go ahead and look at the Lynch paper, which looks at something completely different. There's a reason I'm looking at two very different papers, just so you could see what else you could throw into a results section. So, results starts with QuemScan. Now, what we see here is as shown in Figure 6a. Now, just the way this was printed, Figure 6a is on the previous page. There was too much to fit on one page, so here's Figure 6. Now, so you can see more. You can see there are two parts to Figure 6. There's this pretty picture down at the bottom, which was done by QuemScan. It's a mineralogical analysis tool. And so you can see the pictures that were taken of the minerals. But also up above is the assay that is done with QuemScan, which is basically a mineralogical assay technique. Zoom in and let's look at 6a. So this is a very, very different sort of graph than we saw in the previous paper. Here's 100% of what we saw. What percent is each sort of mineral? PV5 has a lot of this light blue, which is calcite. PV1 has less than half as much of calcite. PV1 has a lot of this fluorescent gray, that's chlorite, compared to these other ones. So here you can quick see, PV1 is green and brown, chlorite and kaolinite. PV5 is mostly calcite, right? You can just quick see what that is. Now, if you go back to the QuemScan paragraph, what you see is the key phyllosilicate groups identified are kaolinite, chlorinite, and illite muscovite. Phyllosilicates are most abundant near the rim of the basin at PV1. So remember, there were 4 different sites where they comprise over 73% of the clay fraction. So what does that say? Phyllosilicates are a class of minerals. In the graph, we were looking at individual minerals. So they were saying, okay, now let's take all of those things and let's just clump them into groups. 73% of what we saw at PV1 were phyllosilicates. It's summing up the graph in a way that interprets it and makes it easier to understand. Now, XRD, X-ray diffraction, okay? There are a number of different ways that this is represented. First is this weird little table. And since I made this table, I can say it was kind of a pain to figure out how to do it. We had spectra that told us approximately how much there was, but it didn't give us actual numbers. So we couldn't put numbers in the table. How did we do it? Well, we just said, let's give relative amounts. If it was a little bit, if we just barely saw it, we'd get a check. If there's more, we'd get 2 checks. And if there was a lot of it, it would get 3 checks, right? ND means not detected. Okay, so that's one way to do it. We have a whole bunch of different factors, all in one table, lots of minerals, several sites, several depths within the site. Are there other ways we could show that? Sure. Here's an actual spectrum. This is what came out of the XRD when I ran it. And so you can see here are the peaks that we detected. Here's a peak that has so much gypsum that everything else is really tiny. What else do we have in here? We have, first of all, cation and anion data. Then we have infrared spectrum. Okay, and you can see there's a reference spectrum, so you can tell which peaks belong to what. And then spectra for all the different sites that we looked at. Here is the interpretation from the near-infrared spectroscopy that we did, invisible. And you can see that they're just going through and saying, okay, we see an absorption at these wavelengths. Here's what they could be due to. Here's some of that data represented in different ways. Here now we're actually plotting it by site, right? 1, 2, 3, 4, 5. And what they've done is for each of those little dips, they've said, well, how deep did it absorb? And you can see the trends that change as you go from one site to the next. So there are a lot of different ways you can represent your data pictorially. Lots of ways you can graph things to show raw data, to show trends, etc., etc. And you can be very, very creative in how you display your data. But we also need to explain each of the figures that we make, not just by going through the data and reading back the numbers to the reader, but rather by explaining trends and relationships. We don't do too much analysis. We don't do comparisons with other people. That stuff should be reserved for the discussion, which is what we're going to be talking about obviously next. That's the one major section we haven't covered. Basically, what you're doing, you're telling the story of your data. Be conscientious about how you design your results section, how you pick your illustrations, how you pick your words. Hope this was helpful, and I'll see you again soon.